here with Dr. Jen, and we're just going to do a quick talk about EA2. Sorry, Dr. Jen, what were you just asking?
I was asking if you would oppose to my showing some slides.
No,
or you prefer that we keep everything, just conversational. We can
that's totally fine. You can go ahead and share. That's fine.
Okay.
Yeah.
I mean, just to give you an overview, it may be a little easier if we do it this way. I don't know if you can see this.
Yes, I can see it.
Okay.
Courtney, can you see it?
Can you see?
Oh, good.
Wonderful. Okay. So this is a snapshot of where Mount Sinai is. So this dark high rise is actually where the main campus is. And it took me some time to get used to it. I was recruited to Mount Sinai in New York City from UCLA in late 2017. And I used, on the campus of UCLA, I used to have to walk from building to building to different labs. But here, I just have to take the elevator to a different floor, and I'm in a totally different department. So it was fun. It's been fun. But I am in the Department of Neurology, and we have a division of Neurotology and Neurogenetics that I was recruited. Oh, sorry.
I just had one question. Otology means something to do with the ears, right?
Yes. So I have a special interest in the neurology of balance and hearing. And
so like the vestibular system and stuff like that.
So yes, the vestibular system. And the truth is, it's not, so we require the collaboration between the vestibular system, the cerebellum, and also the somatosensory input from our feet, from our, from the periphery, for all the information to be integrated in the brain to maintain balance. So the truth is, the main part of my research interest has been ataxia, and I'm preaching to the choir. So basically saying ataxia, you all know that it just means incoordination, and it derived from Greek for without order. And so you are all familiar with these core symptoms and signs. And I just want to emphasize to you that as a neurotologist, I'm especially interested in using abnormal eye movement to understand brain dysfunction. And as you know, ataxia can have many different causes. And so we refer to the primary causes that would encompass all the hereditary forms of ataxia. And then there are also other causes that we call secondary, that is a consequence of other, for example, traumatic injury, infection, nutritional deficiency, immune-mediated, and there are also vascular injury to the cerebellum or to the vestibular system that can cause this. And so here it's a more detailed explanation of what neurotology is. And so we collaborate a lot with neuro-ophthalmology, and so we both, so what is shared between neurotology and neuro-ophthalmology is that we're very attuned to using eye movement as a window to the brain function. And so I don't want to belabor this, but just to share with you, the take-home message is that there are far fewer episodic ataxia syndromes that have been genetically defined than the other hereditary ataxia syndromes, the dominantly inherited and the recessively inherited spinocerebellar ataxia syndromes. And so far to date, there are only 8 genetic loci that have been designated, and there are other types of episodic ataxia that are still being characterized, but episodic ataxia appears to be far rarer than ataxia in general. And so, for example, for episodic ataxia type 8, only one family has been identified so far. So the genetic locus remains to be further confirmed. So episodic ataxia, it just means, I think that for us to recognize this as a syndrome, we're basically looking at patients who, instead of presenting with a progressive decline in their function, that they can actually present with very prominent but distinctive attacks, where the onset and the resolution of each episode is actually clearly defined for the patient. So the patient is perfectly aware, and this is not the usual fluctuating symptoms that we can see in ataxia. And these are actually actual attacks, where the patient may be fine one moment, and then the next moment they know that they're in an attack that can last for variable lengths, depending on the subtype. And usually the trigger is something that can be recognized. And the most common across all the different subtypes include emotional stress, whether it's happy or sad, and also physical exertion. So fatigue as a consequence of physical exertion is also a common trigger. And so listed here are the major types, and episodic ataxia type 2 is the most common.
Right.
So if I just wanted to show you how the abnormal eye movements that we can see. And I don't know how well you can see here if you can appreciate a downward jerky eye movement. Sorry about the camera work. And it's more obvious on left gaze, if you can see that. And so this patient actually turned out to have spinal cerebellar ataxia type 6, but this is in the same gene as for episodic ataxia type 2. So on the left of the slide is a video of a patient with genetically confirmed episodic ataxia type 2. And you can see a lot of wandering. The eyes are just moving around. Right. And when the eye, with gaze to the left, you can see left beating nystagmus. And with gaze to the right, you can see right beating nystagmus. So this we call gaze-evoked nystagmus. And upward gaze, the eyes are pretty stable, but there was a few beats of downbeat. And then on down gaze, it's definitely more apparent. And so these are the classic presentations of abnormal eye movement. And so this is a very, very brief summary overview that 3 of the 8 episodic ataxia syndromes actually result from specific mutations in proteins that are important in ion transport. So these are membrane proteins. And for episodic ataxia type 2, it's a voltage-gated calcium channel. And this is a 2-dimensional diagram of the channel structure. And for episodic ataxia type 1, it's a potassium channel. And you can recognize how it just looks like one of the 4 modules that functional domains that we see in the voltage-gated calcium channel. And then we also have for episodic ataxia type 6, this is caused by mutations in a glutamate transporter. And of course, glutamate is the major excitatory neurotransmitter in the brain. And this is a diagram that we made a long time ago, basically putting together, the potassium channel that allows that's selectively permeable to potassium. And we have the calcium channel that's selectively permeable to calcium. And then we have the transporters that would clear the glutamate that can be released into the synaptic cleft to terminate a neurotransmission. So here's a presynaptic neuron, and here's a representation of a postsynaptic synapse, and this is a diagram of the glia where there are also glutamate transporters that can actually help remove the glutamate from the synaptic cleft. And so the purpose of this simple diagram is to demonstrate that disruption of any of the components can actually cause dysfunction of the neuronal signaling. And I highlighted the proteins that are important for episodic ataxia, but this is the same gene that can also manifest with hemiplegic migraine and also progressive ataxia, as in spinocerebellar ataxia type 2. And so that's it. That's it for my brief overview. And what we're doing is basically we are working on tabulating, basically continuing to track patient natural history. The goal is to improve patient function, patient quality of life, and we're looking to try to understand how mutations actually lead to the manifestation of the symptoms. Right. And in the case of episodic ataxia type 2, the story gets more and more complicated, that we now recognize that patients can actually manifest with epileptic encephalopathy. They can manifest with congenital eye movement abnormalities, and patients can have learning disabilities, in addition to the well-recognized episodic ataxia, hemiplegic migraine, and progressive ataxia. And so how we can tease out the neuroanatomical basis for that, and how to best help patients manage their symptoms is one of the major focus of our work. So I would like to, after that brief introduction, I want to open to questions and comments from everybody.
Sure. Just before we unmute everyone, I had two questions that came up. So you mentioned that I saw in your chart there that almost all of the subtypes of EA are autosomal dominant, right?
Yes.
But I know a few people that have EA that don't seem to have any family history. Wouldn't that kid recessive or something?
So good question. And I think that this is a major challenge, right, for geneticists. So for a patient with no family history, depending on the age of onset, if we have the privilege of also having access to the patient's parents, then we can actually figure out whether or not the patient has new mutations, novel mutations, or whether the patient may have a recessive disorder. So it's very challenging when the onset of symptoms is later in life, and the parents have passed away, and the patient's children are too young to manifest symptoms, or if there's no other relative that we can, track down the symptoms. And so, for example, the patient who led us to the discovery of this glutamate transporter, the gene encoding the glutamate transporter as the genetic locus for episodic ataxia type 6, was a child. So he presented with very, very distinctive episodic ataxia, but he also had other features that did not fit with the known episodic ataxia syndromes at the time. And so we did a simple experiment and looked at his parents and looked at his blood And we screened a few candidate genes. We were just very lucky that one of the genes that we looked at was this glutamate transporter. And we didn't even do exome sequencing. That was like 20 years ago. And we were able to identify a specific spontaneous mutation. So it was just by accident that the child had a mutation, and that neither parents had. And so we were able to perform functional studies to demonstrate that that was important. And subsequently, our work was confirmed by the identification of other patients with similar manifestations who also have different types of mutations in that gene. So that's why it is now firmly established as a separate entity. And also, you may be aware that there was a recent study, maybe published 2 years ago, from the UK and also from Australia. This is on idiopathic or late-onset ataxia. So these are patients with no family history, right. But they have no other cause that can be identified to account for their ataxia. And what was discovered was that there was a very unusual expansion or unstable intronic, so in the non-coding region, that is shared among all these patients that are not known to be related to each other. So there seems to be a founder mutation that can account for these rare manifestations. And so I think that the inspiration to all of us is that we just have to keep looking. And we now have, we actually have the tools to look at the whole genome nowadays. And so an estimated 50% of patients with suspected hereditary ataxia do not yet have a genetic confirmation. And so we've got to keep looking. There must be other clues that we're not seeing right now.
Right. So from my understanding, in other cases of other diseases, all the elements of the disease may be present in an individual, but don't become activated until like some environmental trigger or some other traumatic event, or maybe they're undergoing a lot of stress. Is that something that happens with EA2 or other genetic diseases?
So I think that the challenge or the typical presentation, you're asking a very good question, and it's taking me some time to put together my thoughts. In that, for those who actually manifest during childhood, it's more clear that they just have attacks, and it's lifelong. And what has been puzzling us, and one area where we're really examining our data, and we're further investigating, are those with late onset episodic ataxia. So there's a cohort that we've been following, and these are patients who manifest with episodes of ataxia in their 60s. So basically, they have been perfectly healthy before then. And then all of a sudden, they started having these episodes. And so it's very, very curious what it is, and the genetic underpinning, why they should manifest with symptoms at so late in life, if there is a genetic predisposition. And but there does not appear to be any shared environmental trigger for their symptoms among our patients with
Right.
late onset episodic ataxia.
Right.
So there's no shared onset of viral illness or gastrointestinal discomfort or anything that we can pinpoint.
Right. Last question, then I'll let everyone else ask theirs. So I have heard that although like pretty much across the board, people with ataxia are going to have cerebellar atrophy, right? But I have heard that something like 50% of people with EA2 can get they can show no signs of that, right? Is that for
So
just
this is
EA2?
very good question. So this is something that I think that we definitely need a larger cohort to track, and in fact, I think that cerebellar atrophy is actually quite evident in a large proportion of patients with episodic ataxia type 2. It's episodic ataxia type 1 where we see less evidence of cerebellar degeneration. And but we don't know, I cannot tell you the trajectory or the rate of atrophy. We definitely need, this is definitely an area that we would like to have a better understanding. And episodic ataxia type 2 is interesting in the sense that it really spans the entire lifespan. So we have patients who are newborns, who are infants and toddlers that are manifesting their symptoms. And we also have patients presenting in their, young adulthood with symptoms that have been bothersome for a long time, or become more prominent later in life. And so one question that we're trying to address is that, is this a neurodevelopmental problem, or is this a neurodegenerative problem, or is it both? Because in children, at birth, it's very difficult to pinpoint what the problem is. But over time, we can actually see more clearly signs of cerebellar atrophy.
Right. Okay, so now I'll open it up to questions, but since we have so many people, I don't want to unmute everyone. So kindly hit the reaction button and raise your hand, or just type your question in the chat box and I'll ask it for you. But the first question is from Kristen. Hi, Kristen.
Hi, Dr. Jen. Thank you.
Hi.
So both my young daughter and I have EA2 that's genetically confirmed. We found it because she had symptoms from such a young age, and even as young as age 2, and they couldn't figure out what it was, said it was probably a channelopathy of some kind, but nothing that they recognized, and then eventually did full exome sequencing and found that she has a mutation, and it aligns with EA2, and then found that same mutation on me. And I've had symptoms my whole life, but nobody knew what it was, of course. And now my symptoms are getting progressively worse. I'm not having episodes so much as having this progression. So, and the doctors where we live are having a really hard time sort of filtering this out between EA2 and SCA6 and making this determination. And the complicating factor is I also have this dystonia, and they tell me, well, that's not part of your disease, but I have it, and it's so I can't. I'm like, well, I don't know, but
You didn't read the textbook, did you?
Yeah, so I'm wondering if you can speak to that a little bit more, because I have to be on medication for that. I have to be on Keppra, or I just can't really function.
So I can assure you that it can be a part of the manifestation.
Okay.
I can assure
Thank
you.
you.
So,
Okay.
so we see that. And
Okay.
and sometimes it's very difficult for patients to explain their symptoms, and the findings can be very subtle. But I think that we're definitely recognizing dystonia as
Very
a
helpful.
part of as a core feature of episodic ataxia. Okay.
Thank you. That's very
And
helpful. I didn't
yeah,
even know what it was called. I was just explaining what was happening.
so you feel a tightness, what do you
Yes.
feel?
Yes. And I can tell before it's going to happen. I'll get a weird sensation that something is going to happen, and then I'll feel a zing where it's going to happen, almost like an electrical zing somewhere. And even then, still sometimes I won't recognize it. And then it will, like if say if it's my leg, it will tighten and clench up, and then it may jerk, but not huge jerks, just like little ones. And it maybe lasts for 5-10 minutes, and then I'm exhausted afterwards. And then so the Keppra takes care of that. Like I don't have it, but even with the Keppra, I still have these little like flutters that you can see like in my arm or my leg, and it'll just flutter like that.
So
Strange.
that I think that that's also something that we do recognize, and so this calcium channel that we're that is responsible for that is affected in this condition, it's expressed abundantly in the cerebellum and also throughout the brain, and also it is the most important presynaptic calcium channel at the neuromuscular junction.
Okay.
And so you can imagine if there's any disturbance or any dysfunction of the channel, it can also interfere with peripheral muscular function.
Okay.
Okay.
Doctor,
Thank you.
when you say a channel, do you mean that it transports calcium to wherever it's needed? Is that what it means?
So when I say channel, I mean a protein that has selective permeability for specific ions. And we name these channels based on the ion that they select for. So for example, potassium channel, it has selective permeability for potassium, and calcium channel has selective permeability for calcium. And these channels are fascinating in that they are turned on or off in response to the membrane potential. So they are voltage gated. And there are also channels that are turned like opened or closed, depending on the ligand or something that activates it. And so an example is a glutamate channel, right? Or a acetylcholine receptor channel. And so those channels are more mixed. Once they open, they're gigantic. They allow everything through. But the voltage gated ion channels are super neat. They're turned on depending on the voltage, and they allow, they're just so exquisitely complex and agile in their function. So to be very selective.
Yeah.
And so the thing is that to excite a cell, so to activate a neuron, it's a complex cascade of events where the activation of so the release of of acetylcholine or the binding of acetylcholine to the receptors would allow ions to come through, and would allow the activation of sodium channels. Then a lot of sodium current gushes into the cell, and then the potassium channels are activated. Potassium exits the cell. So it's an careful orchestration of many different ion channels. And so you can imagine that any timing problem or amplitude in the signaling or whatever can cause a problem. So think of it. I actually like the analogy of looking at everything as an orchestra. So if one part is not working, then you get this, horrible cacophony. Okay, that's the problem.
I'm laughing
That's
because
good.
I literally used an analogy in our last meeting.
Oh, great.
That's helpful.
But so we have a question from Jesse. Go ahead, Jesse.
All right, I was wondering, when you had that slide up with the list of symptoms, and you had one that said acetylamide or something, what does that one refer to?
Oh, so channelopathies refer, this is a word, this is a term that have been coined to refer to neurological or actually medical problems that arise from dysfunction of ion channels. So ion channels are especially important in excitable tissue. So we're talking about the brain, we're talking about the muscle, we're talking about the heart. And so there are different types of channelopathies. And when it comes to neurological channelopathies, the prototypical neurological channelopathy, or the first identified neurological channelopathy, is periodic paralysis. And so this is a condition that presents with paralysis like after a large meal. So usually it runs in the family. It's autosomal dominant. The first few families, it was ridiculous in that they can have a family feast, a celebration, everybody's happy, and they overeat, and then half the people can't move.
Wow.
Okay, so everybody knew. So people knew, and this was from 50 years ago, and people knew that there was something that runs in the family. It was something wrong with the muscles, and then it was just by serendipity that people recognized that acetazolamide can actually prevent such attacks, and so acetazolamide then became the medication of choice for anything that we suspect to be a channelopathy. But now we know that it doesn't work for it's not a panacea. But but acetazolamide has been something that's commonly used whenever we figure that, oh, if it's a channelopathy, let's give it a try. Not that we know how it works, why it works in channelopathies even now. So it's very frustrating. For patients with episodic ataxia type 2, that is the medication of choice in the United States. And I think that in Europe, people are more used to using another medication. It's called 4-aminopyridine, and which is a mixed. It blocks a broad spectrum of potassium channels, and so the reason why we use I put acetazolamide in the table is that many of the patients basically everybody who presents with episodic ataxia, probably the first medication that we would try is acetazolamide. And those who don't at all respond to acetazolamide probably doesn't have episodic ataxia type 2, which is usually very responsive. And but it's also limited because there are patients who are allergic to the medication, they can't tolerate it, and for various reasons, and so we desperately need to develop other effective treatments.
Okay, thank you.
So next we'll go for Ben. Ben, you have a question?
Sure, yes. Thank you, Doctor, for your presentation. That was fantastic. I'm very interested in the idiopathic ataxia studies. I understand you mentioned they are in the UK and Australia. Was that one study that branched to 2 countries or 2 separate studies? And the second part of that question is, has anything similar been done in North America?
So very good question. So these are two separate approaches, two separate patient populations.
Excellent.
And the group in Australia had actually been working on it for a long time, and they were focused on a condition that has a catchy acronym. It's called CANVAS, or cerebellar ataxia, neuronopathy, with vestibular areflexia syndrome. So that's the acronym for CANVAS. Okay, and what it means is that these patients have both central problem of cerebellar ataxia. So they have nystagmus, they have abnormal eye movement, they have ataxia. But on top of it, they have much worse ataxia than just patients and other patients with ataxia, in the sense that they also have no inner ear vestibular function. And so it's a double whammy. And then on top of it, they also have a peripheral neuropathy. So they have, basically, they are devastated. They just have this terrible inability to navigate, okay? Because we rely, so basically our brain rely on input from the periphery, right? We need somatosensory input, and we need proprioception. We need to know where our feet are, right? And then we need to be able to see, and the information will be transmitted to the brain. And then we need to also, we rely on our inner ear to help us balance ourselves. But imagine all three systems are disrupted, and these people are just absolutely devastated. And so the group in Australia actually have been working a lot on this condition. The vast majority of their patients have no family history and they don't have siblings, but a handful of families actually presented with SIB pairs. So they're siblings, very, very rare. And so this syndrome is also difficult to recognize because they don't present all at the same time, with cerebellar ataxia and inner ear dysfunction and peripheral neuropathy. And so the different symptoms can come on at different times, but with the common feature that they just all have terrible balance, okay, terrible imbalance. And so the Australian group were working on this, and because of the absence of a multi-generational transmission, they knew that it's unlikely to be autosomal dominant. And the rare observation of SIB pairs suggested that this could be autosomal recessive. And so they screened for, so they went through the usual, it's definitely not any of the ataxia genes. And then exome sequencing turned up nothing, no viable candidate. And so these people started doing, these researchers did genome sequencing. And so they found a dynamic repeat expansion. It's a pentanucleotide expansion in a gene with unknown function. And around the same time, this British, so UK group was also working on patients with ataxia. And so they were focusing on patients with late-onset ataxia, no family history, presumed idiopathic origin. And they looked at their patient population and found the same pentanucleotide expansion. And so it's fascinating that they also recognize that it seems to be a founder mutation, and the same gene had been screened for in Canada, and also elsewhere in the United States, the findings is that this seems to be a very important gene that accounts for a small fraction of all patients with idiopathic ataxia. The initial presentation, the initial observation in the UK was that it accounted for a large proportion. It's like 20-30% of their patient population, which is a huge amount. And but probably it's because the patient cohort may be more ethnically homogeneous in the UK compared to the population cohorts in North America, in Canada, and also in the United States. And but very interesting question. Thank you for asking for more details. I appreciate your curiosity.
No, thank you for the elaborate answer. It's quite good. Thank you.
Okay, I have 4 more questions. I'm going to ask 2 more quick ones, and then we'll take 2 more from the group. So, Dr. Jen, first question. Since you mentioned that group in Australia, they specialize in canvas.
Yes.
Now there's one person I see here, Gwen, who has canvas, and I believe one of my other friends. What is the name of that group? So maybe they can reach out to them.
Absolutely. Let me look, and in the meantime, you can ask me another question.
Okay.
How's that?
So sure. Do you know anything about RSACs? It is a cerebellar ataxia.
Sure. Just the basic things, yes.
Oh,
Why?
Oh, because someone also had a question about that. What is the best advice to help someone with RSACs? But I don't know if you would know the answer to that. No.
Well, it depends on what your question is, because right now our if you're asking, what can we do about it, or what can we, if you're asking about rehabilitation or symptomatic relief or whatever, what we do know is that there is so we don't have a gene-specific therapy yet, right? The goal is for all the hereditary ataxia syndromes is that we actually have some kind of gene-specific therapy that can be effective, right?
Right.
And so I'm very optimistic that will become a reality, because we now can edit genes, we can block genes. There are different things that we can do, but there's at this time no specific treatment yet.
Okay, so did you find that information on the show?
Yes, I would. So I'm going to put in the comment, and because the neurologist who was involved in delineating this syndrome, sorry, and let me.
That's great. And I'll put that in the sort of the follow-up for everyone to look that up as well.
He was the one, because prior to him, I think that for years, everybody recognized that there is an unusual combination in exceptionally rare patients to have both vestibular areflexia, so loss of inner ear function, and cerebellar ataxia. But it was Dr. Schmelwitz who actually figured out that these patients also have a very unusual peripheral neuropathy, and that the vestibular areflexia was a part of the manifestation. So they did incredible pathological characterization and symptomatic characterization, and then he was involved in the genetic investigation, So hats off to him. So I think that I'm sure that he would be so informative if you were to
Great.
ask him for questions.
Okay, we'll take one question from David and one from Charlotte. So David, you can unmute. Go ahead and ask your one question, David.
One?
Yeah.
No!
One.
Just one? Ah, you're cruel. Ah. I got to rearrange my mind now to think of which one I want to ask. Do AE1 and AE2 mix over? And on your list of symptoms, it says no. When it says no, does that mean, well, maybe no? Or how definite are you guys with that on your symptoms? I mean, could AE1 still have tinnitus and AE2 still have tinnitus?
So the
when-
yes, no, usual, occasional, I think that these are just as a general rule of thumb. It's by no means definitive. And as you know, we were combining information from different patients, and so this is just a very rough estimate. But if a patient were so all that I'm saying is that if you were to look at this and then you have a patient with many of the features and then they have something that's out of the ordinary for this particular syndrome, that alone would not disqualify the patient for a specific diagnosis, obviously, right? Everybody, I mean, by now we recognize that there's always clinical heterogeneity, but it's just that overall, this is the best overall picture that we have. It's a generalization, we recognize that, but it serves as a good guideline clinically. It's convenient, but I would definitely go by the genetic diagnosis, and now that we have genetic characterization, that we absolutely require definition. So earlier, Kristen said that she was genetically confirmed, because you'd be surprised. 20 years ago, a lot of people were carrying the diagnosis of episodic ataxia too, without genetic confirmation. And it's just that they were ataxic and they had nystagmus, but that's not specific. And so I think that those people, it would be best to say that they have EA2-like features that await genetic confirmation. And but once you have the genetic confirmation, and then you have certain features. So the thing is, it's very, I want to caution that we shouldn't jump to conclusions. If a patient were to have a genetic abnormality and he has a million features, we can't easily attribute everything to that mutation, because it's possible that he has other conditions, right? And so we can only come to a conclusion defining the clinical spectrum of a specific condition if we have information from more people, a cohort or large series of patients.
Great. And last question is for Charla. Go ahead, Charla.
Okay. I guess, is there best alternative therapies without medication for sleep therapy for one with leg tremors and wandering mind?
You are a tough crowd. You ask some very questions that I can't just give you a snappy answer.
Is there a forum we can start?
So no, this is very important. I think that your question also exposes our ignorance about the importance of sleep. I think that our society undervalues sleep. So there are all kinds of ads about what we can take to keep us awake. But we don't value natural sleep as we should, and I think that a lot of the we're beginning to recognize that a lot of the neurological conditions can definitely be correlated with sleep disturbances, and so that's a very important question that you raise. So I think that anytime it depends on our priority, and it depends on what the problem is. And we recognize that, something like restless leg syndrome, that if a person without medication simply cannot sleep, and the legs are just, so bothersome, and the urge to move is so uncomfortable, and it's overwhelming. I think in that circumstance, we definitely need, I think that the patient can use some rest, and that it would be helpful to provide some relief. And so it doesn't mean that we have to jump right to, clonazepam or, another benzodiazepine or other medications. And we do recommend exercise. We do recommend, double checking the ferritin level. We do recommend on meditation. And, there are other things that we can do. And in fact, I do appreciate the question, because lifestyle is so important. What we eat and what we do and how we live and everything. And so, yes, I'm very supportive when patients would like to try a non-pharmacological intervention. And I think that we also have to have a long view to improve patient quality of life with minimal side effects.
Right. So before I let you go, Dr. Jen, there are probably some other questions that we didn't get to. How can anyone reach you or with other questions about EA2 or any?
So I'm going to type my email here.
Okay.
And this is how Courtney found me. And I'm sorry that I recognize that my answers are all long-winded. I wish that I could be more brief.
We all understand that this problem is very complicated, so it's hard to give a yes/no answer for anything.
But thank you for having me, and thank you for making time. I appreciate your questions. I appreciate your curiosity, and you are who inspire me to find solutions and try to be of help.
Yeah.
Thank
Okay.
you so much for your time.
Thank you so much.
Yeah.
Thank you.
Nice meeting you. Bye bye.
Thank you.
Bye.
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 Perfect. Thanks.
Bye bye.
What do you think?